Hi, this is lesson number 33 and it's entitled Critical Process Parameters, or CPPs as they're commonly known. The objectives of this lesson are as follows. I will cover some of the definitions associated with CPPs and explain their relationship with CQAs, or critical quality attributes. CPPs will be explained in more detail, along with how you could define them for a process and how they're selected as part of the process design. The regulatory requirements are discussed, and the final slide includes some references that are required, some additional reading around CPPs and the regulations associated with them. The first thing we need to talk about when we're talking about CPPs is their relationship with the CQAs, or the critical quality attributes of your process. The industry generally associates critical quality attributes with the ICH parameters such as identity, purity, potency, etc. CQAs are the foundation upon which CPPs must be associated. So line of sight between CPPs and CQAs is considered a major component of the drug development strategy. Now, the ICH definition of a CQA is a physical, chemical, biological, or microbiological property or characteristic that should be written within an appropriate limit, range, or distribution to ensure the desired product quality. Again, they're considered highly critical during the drug development stage. The CPPs then, the definition is a process parameter whose variability has an impact on a CQA and therefore should be monitored or controlled to ensure the process produces the desired quality. And that's the ICH Q8 definition. So CPPs are attributes that are monitored to detect deviations in standardized production operations and product output quality, or changes in critical quality attributes. The control of critical process parameters during processing is the basic need of a validated process. If critical parameters are not controlled, it will contribute to the variability of the end product. It should happen during the drug development stage, or the design stage, where critical parameters and in-process controls are identified. A process parameter whose variability has an impact on a critical quality attribute and therefore should be monitored or controlled to ensure the process produces the desired quality. Again, a definition from the ICH Q8 revision 2. CPPs have a direct impact on the critical quality attributes. A process parameter, or a PP, can be measured and controlled or adjusted, and some examples of CPPs for small molecule are things like temperature, addition rate, cooling rate, rotation speed. Examples of CPPs for large molecule are again temperature, pH, agitation, dissolved oxygen, feed type and rate, medium constituents, etc. When is a PP, or a process parameter, considered to be a critical process parameter? Identification of all unit operations and their associated equipment sets and equipment capabilities in upstream and downstream processes are crucial when selecting those parameters that need to be controlled to ensure CQAs are consistently achieved. Small changes in time, temperature, pH, and other variables may result in changes to the API characteristics, or even the yield or the impurity profiles. And because biologics are extremely sensitive to processing, it's important that each unit operation is carefully evaluated for possible impact to the large molecule and the impurities thereof. So CPPs are responsible for ensuring the right CQAs are met, and CPPs are identified from a list of potential processing parameters using risk assessment and experimental work during the drug development stage. One method of defining CPPs is to look at the effect of certain production processes on critical quality attributes. For example, drying time in a dryer on LOD values for a small molecule API powder. Those production parameters that have a measurable effect on product quality should be tightly controlled and are considered to be a CPP in your process. Manufacturers should conduct tests to set acceptable range limits of the CPPs, and they should determine acceptable process variability ranges. Operational conditions within the range is considered to be acceptable. Any deviation outside acceptable range is indicative of issues within the process, and thus the production of substandard products or out of specification products. Data relating to CPP should be recorded, stored and analysed by the manufacturer, and historical CPP data should be reviewed carefully to detect trends or drifts. Selecting CPPs then. So the key steps to selecting CPPs and their application to process control are as follows. Identify critical quality attributes or CQAs for drug product and drug substances. Select API excipients, materials and container closures. Materials and container closures, the key parameters and analytical methods that measure the attributes of the APIs or the key materials, packaging, container closures, have to be examined during quality risk management or during QRM approaches. This approach focuses on finding those attributes that would be critical to maintain the quality and stability of the drug substance and drug product. Key findings of this QRM, quality risk management review, are added to the CPP list that needs to be controlled. The next step, to define all unit operations and process flow. Identification of all unit operations and their associated equipment sets and equipment capabilities, both in upstream and downstream processing, are crucial when selecting those parameters that need to be controlled tightly. Small changes in temperature or pH can have a huge impact on your final API specifications, namely your yield as well as your impurity profiles. Define all product and process specification limits. Specification limits for the product and its process must be defined to protect your CQAs of the drug substance and your drug product. Specification limits will form a key basis for CPP determination. Specification limits are primarily defined for product control rather than for process control, but they're still very important to assess as part of your QRM, quality risk management process. Achieve acceptable results for method validation of all analytical methods. So limit of detection, limit of quantification, precision and accuracy must be characterized for all analytical methods, and method validation must be completed. Once these steps are done, you can then trust the numbers and know the error associated with any results coming from the QC lab. Method validation should be done prior to product and process characterization studies and the design and implementation of process control. Complete quality risk management for factor response selection for all critical unit operations and materials. A formal QRM process should be in place to systematically examine all materials and unit operations for their potential influence on the CQAs. Risk ranking and other QRM tools are used to identify factors and unit operations that hold the greatest risk. Scientific understanding and historical data are typically the basis upon which potential risks are identified and prioritized. CPPs may be identified in this process that will later need to be ruled in or out based on data and identified risk. So explore the design space for all key factors identified during the risk assessment using design of experiment DOE or other multivariate methods. Determine the factor effect size and select all CPPs. Evaluate the CPPs for ease of control and practical application to process control. This slide looks at some of the regulatory requirements around CPP selection. So the ICHQ7, one of the definitions says in the validation section, defining the APIs in terms of critical product attributes. ICHQ8, a definition taken from it is, at a minimum, those aspects of drug substances that are critical to product quality should be determined and controlled strategies justified. And finally, ICHQ8, manufacturing process development should include at a minimum the following elements: identifying potential CQAs associated with the drug substance. The systematic approach for CPP selection and use must fall in line with the International Conference of Harmonization Q8, 9, 10 and 11 guidelines, which recommends quality risk management and the identification of CPPs as part of drug quality and process control development. And finally, this slide includes some of those reference documents that I've spoken about. Again, it would be of huge value to go in and look through those documents to get a better feel for CPPs and CQAs and the relationship between both. And that's the last slide. Thank you for listening.